Hello, you're welcome to our lecture series on biological techniques. Earlier in this lecture series, we had looked at the introductory part of microscopy, the major types of microscopes, and we had also looked at how to prepare microscope slides. In today's lecture, we'll look at the types of the compound optical or light microscope. Now, what is the compound optical light microscope? The compound optical or light microscope. The compound optical or light microscope is a microscope that employs visible light to produce images. And the visible light could be in form of the ultraviolet light from the sun, or it could also be light from electricity. So we want to look at the different types of the compound light or optical microscopes. The different types of the compound light optical microscopes are: one, we have the bright field microscope, the dark field microscope, the stereoscopic or dissecting microscope, the confocal laser scanning microscope, the fluorescent microscope, the phase contrast microscope, the differential interference contrast microscope, and the inverted microscopes. 8 types of the compound optical or light microscope. So I want to look at the bright field microscope. The bright field microscope has dark object and black background as a very distinctive feature. The compound bright field microscope is the most common type of light microscope available in most institutions and laboratories, especially in biology laboratories. It's common in secondary school laboratories, university laboratories, and even in some primary schools. It is called a bright field microscope because it forms a dark image against a brighter background. It produces a dark image against a brighter background. The image is usually dark, while the background is brighter because of the illumination of light from the bottom that passes through the condenser to the stage. And the compound bright field microscope specimens are visualized because of the slight differences in contrast that exist between them and the surrounding medium. With a conventional bright field microscope, light from an incident source is aimed towards a lens beneath the stage called the condenser, through the specimen, through an objective lens, and to the eye through a second magnifying lens. That's called the ocular lens or the eyepiece. Now, this is a bright field microscope commonly used in most laboratories, but this one is more sophisticated, having digital part. I want to look at the uses of the bright field microscope. The bright field microscope is extensively used for viewing microorganisms. Usually, it is necessary to stain the specimen before viewing, but it can also be used to view unstained specimen. Some specimen like bacterial specimen would have to be stained either through gram staining or other forms. But there are some specimens that can be viewed with the bright field microscope without staining. For instance, the protozoans, the bacteria, fungi, and other, the protozoans such as the amoeba, the paramecium, the chlamydomonas, and others can be viewed without being stained. The second type of the compound light microscope we want to talk about is the dark field microscope. The dark field microscope has bright objects, but has a dark background in contrast to the bright field microscope. In a dark field microscope, dark background is produced against which objects are brightly colored. It produces detailed images of living unstained cells and organisms by simply changing the way they are illuminated. It's able to produce details of living unstained cells. So the cells do not need to be stained in a dark field microscope. But a bright field microscope will require staining of some organisms. Now, I want to also look at the fact that all standard bright field microscopes can be readily converted to dark field by inserting a round opaque disc beneath the condenser. So bright field microscopes are easily converted to dark field by inserting opaque disc beneath the condenser. And that opaque disc now creates a dark field that the organisms are now viewed. That is, a dark field microscope uses a dark field condenser that uses an opaque disc instead of the normal condenser. The disc prevents light from entering the condenser directly. The light that enters the objective lens is the one that is reflected off the specimen. The light that enters the objective lens is the one that's reflected off the specimen. Because there is no direct light at the background, the specimen appears to emit light on a black background or dark field. The specimen appears to emit light on a black background or dark field. Now, if you want to have a better understanding of how the dark field microscope works, you compare it with the stars in the universe. The stars are there, but they cannot be seen. But they are seen, they cannot be seen during the day, but they are seen at night. The night appears to be the dark field that's created. So the dark field now makes these stars visible. That's how some microorganisms are. So microorganisms cannot be seen with the bright field. It's by making the field dark that they can also be visible. So that's the purpose that the dark field microscope achieves, to make organisms that cannot easily be seen with the bright field microscope visible. Now I want to look at the uses of the dark field microscope. The dark field microscope was first utilized to examine transfiltrable infectious agents, later to be termed viruses, and to determine that they were particulate in nature. So it's used to view viruses. It was first used to view viruses. Earlier, the viruses were called transfiltrable infectious agents. So these viruses are well properly viewed with the dark field microscope. Small objects, even those below the limits of resolution, can be detected easily with the dark field microscope as well. It is a very simple yet effective technique and well suited for uses involving live and unstained biological samples and microbes, such as a smear from tissue culture or individual waterborne single-celled organisms. Now, the dark field microscope is used to view live and unstained biological samples or microbes. Microbes that are not stained and that are still alive can be easily viewed with the dark field microscope. And it is very effective in viewing such microorganisms. It is also used to identify certain bacteria, such as the thin and distinctively shaped Treponema pallidum, the pathogen that causes syphilis. The pathogen that causes syphilis, Treponema pallidum, is easily being viewed with the dark field microscope. Treponema pallidum is easily viewed. Now, this is the image that's created by the dark field microscope is the image of a bacterial colony. The bacterial colony appears to be bright against a dark background. They appear to be brightly colored amidst a dark background, just like the stars in the skies at night. So it's effective in viewing such organisms that cannot be easily seen. The next type of microscope we want to discuss about is the stereoscopic or dissecting microscope. The stereoscopic or dissecting microscopes. Now, the stereoscopic microscope is also called a stereo microscope for short, and it's also referred to as the dissecting microscope. A stereoscopic or stereo or dissecting microscope is a relatively low magnification microscope that is good for viewing large objects. This microscope is good for viewing large objects. It usually has a very low magnification. It's often from ×2 to ×30. As its name implies, it is better than a compound microscope for dissecting many materials, such as small animals, plants, or organs, because it produces a three-dimensional image. It's used for dissecting. It's used for dissecting, and it produces three-dimensional images, 3D images. Now here is a dissecting microscope. Here's the eyepiece of the microscope, the diopter ring, then the microscope body, the objective lens, the light source, the stage clip, the stage plate, and the base, then the focus node, then the arm. One thing that's very observable here is that the light source, particularly in this dissecting microscope, is from top to bottom, and it has just one objective lens compared to the bright field microscope. Now, uses of the dissecting microscope. What are the uses? A dissecting microscope, also known as the stereo microscope, is called so because it is frequently used in dissecting operations. Its lower magnification ability and long working distance range of 25 to 150 millimeters enables the user to manipulate the small specimens such as insects. So it has a long working distance. The objective is much higher in position than that of the bright field or dark field microscope. So it's easier to manipulate the specimen while working on it, while trying to view it in a dissecting microscope. A dissecting microscope allows the user to observe live specimens as well as perform dissections under the microscope. It is used to view live specimens, and it also enables the user to perform dissection right under the microscope. That's why it's called a dissecting microscope. It's used to dissect small organisms by viewing through them, and it enables one to see exactly how to go about the dissection clearly. A dissecting microscope is used to view three-dimensional objects and larger specimens with a maximum magnification of ×100. Has a maximum magnification of ×100. This type of microscope might be used to study external features on an organism or to examine structures not easily mounted onto flat slides. Now, here is a table that shows a distinction between a dissecting microscope and a compound light microscope. Now, the magnification of the compound light microscope is usually from ×40 to ×100, but a stereo microscope has lesser magnification, usually ×7 to ×40, maximum of ×100. Then the compound microscope do not have a stereoscopic view, while the stereo microscope has a stereoscopic view, a three-dimensional view. The stereo microscope has a three-dimensional view. It can produce three-dimensional images, 3D images. Then the compound microscope, the light microscope, cannot be used to view opaque objects, while the stereo microscope can be used to view opaque objects. Then the depth of field in the compound microscope is low, but is high in the stereo microscope. Illumination is from bottom up. The light shines through the specimen from the bottom in the compound light microscope. But in the case of the dissecting microscope or the stereo microscope, the light source is from bottom up, and it can be from top bottom. Mostly it's from top bottom. Opaque objects can also be viewed with the light shining from the top. So it's used to view organisms from top to bottom. The light source from top to bottom. Now the specimens, the objects that can be caught, the objects that can be caught very thin and still allow light to pass through. The objects that are to be used in the compound light microscope are expected to be caught very thin. Okay, thin slices are made before they are being viewed with the with the compound light microscope. But with the dissecting or stereo microscope, the objects are usually very thick. They're usually thick and opaque. The specimen to be viewed are usually thick and opaque. Now, the compound microscope can be used to view eukaryotic cells. And in fact, virtually all eukaryotic cells can be viewed with the compound light microscope. But the dissecting microscope, though it can also be used, but it has few limitations. It does not have much magnification of as it is in the compound microscope. Then the compound microscope can be used to view prokaryotic cells like bacteria, while the stereo microscope cannot be used to view bacteria. Thank you very much for watching this video. Watch out for subsequent videos in our YouTube channel. In our next video, we'll look at the remaining types of the compound light microscope. Thank you.